So welcome, everybody. Actually, I shouldn't say this, but I noticed in the group that Katerina Manassas has joined us. And I will say, Katerina, first of all, it's been a long time since we saw each other. But I don't know if I personally am keeping your book in sales, but I recommend it to every single parent of anxious kids. So I think it's a well-used and widely respected book. So I'm glad to see you here. Welcome, everybody, again. As we know, so our guest speaker today is Dr. Susanne SCHMID, who is the Vice Dean of Basic Medical Sciences at Schulich, and is fantastic in that position. Susanne, well, I like Susanne a lot, and I think she's great at many, many things. But so Dr. SCHMID is a professor in the Department of Anatomy and Cell Biology at Western, and as I mentioned, is a Vice Dean of Basic Medical Sciences at Schulich. She did a PhD in biology at the University of Tübingen in Germany, and then a postdoc at the University Eye Hospital in Tübingen as well. And then she led her own research group at the university and was awarded a Heisenberg Fellowship, although we're not entirely which Heisenberg is named after. And she spent 1 year in the Department of Psychology at University of Toronto, and then moved to Western in 2007. She has been in a lot of administrative and executive positions at Western over the years. So from 2015 to 2018, Susanne was the director of the Neuroscience Graduate Program, and then was the Associate Dean for Graduate and Postdoctoral Studies at Western and Vice Dean, and now became Vice Dean of Basic Medical Sciences in 2020. But for our interest, Dr. SCHMID's research program focuses on the neural mechanisms of sensory filtering and changes in sensory processing in neurodevelopmental disorders, using environmental and transgenic rat models and a variety of different approaches. And for eons now, I think, has had continuous funding from NSERC and CIHR and other organizations. And apart from being a great administrator and a fantastic scientist, Susanne is also a great person. So, Susanne, thank you very much for joining us. It's wonderful to see you again.
Well, thanks, Rob, for the really kind introduction. I am going to share my slides.
Okay. As we discussed, Susanne, please make sure that there are no tabs showing in other places.
I think I made sure of that. Is everyone, can you see my slides now?
Yes, we can.
Okay, good. Okay, again, thanks for the kind introduction, Rob. And as Rob mentioned, I'm a biologist. So my talk about autism and sensory processing or changes of sensory processing in autism is really from a biological view more than, a physician or a psychiatrist or anyone else. So I jump right in. I think I don't need to introduce much about what autism spectrum disorders are. I just want to emphasize it is a spectrum, and it's, of course, a neurodevelopmental disorder. And we know that it has genetic and environmental causes, or often it's thought to be maybe an interaction of these two. And so when we look at the genetic causes, there's a very small fraction of autism that is caused by one gene mutation or loss of function or gain of function. And these are the so-called syndromic disorders like Rett syndrome, Fragile X, and others. And again, they are relatively rare. But then there are more than 100 risk genes that have been associated with autism or increased risk of being diagnosed with autism. And so these are the genetic causes. And then among the environmental causes, maternal immune activation during pregnancy, so a severe viral or bacterial infection that causes maternal immune activation has been shown to increase the risk for the child to be later diagnosed with autism. We know that drugs like valproic acid, which is an anti-seizure medication, but things like old age of a father and so on are associated with a higher risk for autism. I think of these environmental causes, many of them either directly impact brain development, or often they have an epigenetic effect. So they, to some extent, do influence gene expression. And that could probably include any of these risk genes as well. So speaking of genes, from a biological point of view, so there are these hundreds of genes, or several hundred, few hundred genes that have been associated with a higher risk for autism. And here on this slide, I show a synapse. This is a synapse between two neurons. And there are a lot of different proteins and genes involved in synaptic transmission, in the formation of synapses, and then also in the stabilization and maintenance of these synapses. And all the names here in red are autism risk genes, so genes that are associated with a higher risk for autism. And so I think it becomes evident that from a picture like this, that many of the risk genes for autism actually interact, or their proteins interact. And so from a biological point of view, people think, or I think it's pretty much accepted nowadays, that there might be maybe 5 or 10 different biological processes, signaling cascades, where genes interact that are affected that are affected in autism. And so if any of these signaling cascades are affected, there's a likelihood that this child is diagnosed with autism. So that means from a biological point of view, that many of these risk genes converge on a single signaling pathway, or on a biological process. And then these biological processes are maybe 5 to 10 different ones. So one is synaptic transmission and the formation of and stabilization of synapses. Another would be neural migration during development. Another one could be myelination of axons, and so on and so forth. And a little bit in that line, there were studies using functional MRI, resting state MRI, with, in this case, 16 different mouse lines that all have a knockout or autism-relevant mutation. And when the functional MRI, resting stage MRI, so the connectivity within the brain is analyzed in these 16 mouse lines, they fall into 4 different clusters. And here you see, I don't want to go into detail here, but here you see the 4 clusters and the genes that fall into these clusters. So that again is playing to this idea that there are different types of autism, or there are clusters within autism where similar brain areas or similar pathways are affected. And these build one cluster. And a similar study has been done in humans using resting state fMRI in participants that have been diagnosed with autism, ADHD, or OCD, and that included the POND network, so the Province of Ontario Neurodevelopmental Network, and also the Healthy Brain Network that is based in New York. And these participants underwent functional MRI, resting state MRI scanning, and an analysis was done to cluster them. And they found 6 different clusters that align well with some of the behavioral symptoms, but did not align very well with the psychiatric diagnosis that these individuals actually had received. So what I want to say with that is that from a biological viewpoint, autism includes a number of maybe 5 or 10 basically different disorders where different pathways and biological processes are impacted that lead to behavioral symptoms that then all are come together to an autism diagnosis. But from a biological point of view, it might actually be 5 to 10, somewhere along that, different disorders with each with different severity. And This makes the development of a causal treatment, of course, very difficult, because depending on which pathway is impacted, there might actually be a different causal treatment necessary. And so we don't, in most cases, we can't identify the underlying cause, except maybe in these syndromic cases. And so that makes it difficult. It also makes clinical trials difficult, because, of course, in a clinical trial, if anyone with an autism diagnosis is included, we actually might have participants in a trial that have actually 5 to 10 different disorders all in one group. And if a treatment might help 1 or 2 of these groups, but not all of them, the trial will not show efficacy. So what we probably would need to do is to analyze when and where all these autism risk genes are expressed during human brain development, and see how they interact, and then identify these biological pathways that are disrupted, and develop treatments to really remediate these disrupted pathways. And we need to do this probably early in brain development. And that is, of course, easier said than done. This is almost impossible to do in the human brain. So what is done in the meanwhile is, as Rob mentioned last time, we treat symptoms or a group of symptoms. And I, as an animal researcher, we also focus mostly then on a group of symptoms in order to find out what are actually the mechanisms underlying autism and how can we treat them. So the group of symptoms that we in my group focus on are sensory processing changes. Why changes in sensory processing? Well, first of all, changes in sensory processing are a core symptom of autism spectrum disorders now with the DSM-5. Sensory symptom severity often predict other symptom severity. And that means that at the very least, there's a common molecular mechanism. So whatever biological pathway might be affected might not only affect the sensory pathways, but also higher cognitive areas and so on. So when we learn something about how sensory processing is disrupted, we learn something about what general pathway is disrupted in the brain, that also then impacts higher cognitive function, social behavior, and so on. But there's also a little bit of a debate to what extent sensory symptoms actually cause other symptoms. And to elaborate a little bit on that, you have to be aware that the only input into the brain is sensory information. That's all that goes into the brain. And then the brain stores that, processes it, compares it with experience, and so on. And the output of the brain is motor commands that create behavior. And so a lot of this wiring in the brain, how sensory information is relayed to the cortex and then to other cortices, is kind of hardwired, develops during brain development. But then there is in all sensory pathways something that we call a critical period during brain development. And this critical period is after the onset of sensory input. So when the eyes are opening, when the ears are open, when they start to feel, get tactile stimuli, and so on. And in this critical period, there's a very high plasticity in the brain that fine-tunes the neuronal connections. So we know that these, retinotopic and tonotopic maps in the cortex get fine-tuned during input-dependent. We know that, for instance, half of the retinal ganglion cells, which are the cells in the retina and the eye that form the optic nerve, are actually undergoing cell death during this critical period because they're not properly connected. And then the remaining ones, they form the fine-tuned functional connections. So there is this critical period of high plasticity. And the idea is if the sensory input is distorted, during this critical period, this has a major impact, a ripple effect on other areas in the brain, and it really changes the development, the trajectory of brain development. And so there is this discussion to what extent sensory issues, sensory processing changes are really having a large impact on brain development. The good thing is, and that's one of the reasons why we also look at sensory pathways, is they are well described and they are relatively good accessible for measurements, and they are highly conserved. So between across animal species, but for sure within vertebrates and mammals, the sensory pathways are almost the same. So they are really highly conserved, which gives a great opportunity for study disease mechanism and to test therapies in animal models when we look specifically at the sensory pathways. And then we hope to learn more about the biological processes that are involved in a certain phenotype. So what animal model do we actually use? Because there is, of course, no autistic animal. But what the animal model that we use most in my group, we use different types, but lately the most prominent one we use is the catnap2 knockout rat. So that is a rat that has a complete loss of function of the catnap2 gene that codes for a protein called Caspr2. That gene has been identified in the Amish population, old order Amish population in Pennsylvania, where there is a very high incidence of a severe form of neurodevelopmental disorder called cortical dysplasia focal epilepsy syndrome, CDFE, which is caused by this complete loss of function of the catnap2 gene. It was firstly described in this population where there are families where half of the kids have this syndrome. So it is one of these rare single gene causes for a neurodevelopmental disorder that presents with core symptoms of autism. So these kids have disruptions in social behavior. They have stereotypic movements. They are cognitively impaired. They start to develop language and then regress and end up nonverbal. And relating to the last talk of Rob, they also show increased aggression. And then they get seizures, increasingly seizures. And our rat shows a lot of these phenotypes, sadly, including the seizures as they become a little bit older. Catnap2, we know, is expressed heavily during development and mostly in sensory pathways, and then in humans in the prefrontal cortex. The prefrontal cortex of humans is not well represented in rodents. They do have a prefrontal cortex, which is very small and does not really have this high cognitive function as it has in humans. This is why another reason why we focus more on the sensory pathways. Apart from that complete loss of function of catnap2, there are other mutations in catnap2 and genetic variants, like single nucleotide variants, that have been associated with a higher risk of autism. And then most importantly, as I already mentioned, the knockout rat and the mouse show core symptoms of ASD, including the social behavior disruptions. They have interesting communication, they have stereotypic behavior, they are hyperlocomotive, they also get seizures and so on. So just a little bit about the gene, very briefly. The gene catnap2 is, I think, the biggest human gene, or at least among the biggest. It's located on the long arm of the 7th chromosome. It is a transmembrane protein with a transmembrane domain, a small C-terminal in the cell with a PTZ domain, and a huge N-terminal, which is on the extracellular side. And what you see on the top are all these different mutations, and all the ones in green, and there are deletions here below, all the ones in green lead to or have led to an autism diagnosis, the blue ones to intellectual disability, CDFE, the purple one, speech delay, ADHD, epilepsy, and here in gray, in brown, the schizophrenia diagnosis. So this protein that is coded by this gene, Caspar2, is a cell adhesion molecule and receptor. It mediates interactions between neurons and between neurons and glia during development. It's involved in myelination of axons, but also in the localization of potassium channels within differentiating axons. It stabilizes synapses between different cell types. And it is highly homologous with between rodents and humans, actually to an astonishing degree. So we thought this is a great model to work with, to look into sensory processing. And within sensory processing, we focus mostly on auditory processing because auditory processing is easy to control in the lab. There are a lot of different electrophysiological and behavioral readouts at different levels of processing that we can apply in both humans and rodents. And I want to quickly go into these different measures that we apply. So one of the measures is the auditory brainstem response. That is a neuronal response to sound. And we can measure this as a waveform here. And each peak of the waveform refers to the neural signal traveling through these different stations of the ascending auditory pathway here on its way to the thalamus and the cortex. So we see different stations here in the brainstem, and each waveform here corresponds to the signal passing through these different neuronal substrates. We also can measure an auditory evoked potential with an EEG. This is a signal that the sound creates here in the auditory cortex that we can easily measure. And then there are behavioral readouts. We can measure the acoustic startle response, which is a motor response in response to a sudden loud noise. And the input into the startle pathway, which is a very short pathway, only including the brainstem, the input comes here through the cochlear root neurons and the cochlea neurons. So it's the same as we measure with the auditory brainstem response, the first two waves. But then these cochlea neurons also make synapses in the reticular formation in the brainstem on these what we call PNC neurons that directly project then to motor neurons in the spinal cord that make the animal startle. So the startle is a motor response. It's a readout of this very early processing of auditory sound, acoustic sound here in the brainstem. We can measure prepulse inhibition of startle, which is an inhibition of startle by a preceding stimulus, and we know that the preceding stimulus activates a feed-forward inhibition that includes the midbrain here, the superior and the inferior colliculus. So by measuring prepulse inhibition, we get some information about potential changes here in the midbrain. And then, of course, we can have all types of perceptual tasks where we can ask what is loud, or we can ask to rate fast and slow repetitions and things like that. The nice thing about all these measures is that we can do that in humans and animals alike. And this is work from Kayla Scott, a former PhD student, and Ala Saif, who is a current PhD student. Kayla measured this in rats, and Ala is measuring this in participants with and without autism and ADHD. And so we can measure these cortical evoked potentials, which are just cortical responses to sound. We can measure the auditory brainstem response. And again, these are just electrodes glued to the outside of the scalp and then a sound displayed to a kid while they interact, for instance, with something quiet on an iPad or so. And we can measure this in rodents. We normally measure this in anesthetized rodents. by just inserting electrodes underneath the skin onto the scalp. And as you can see, the auditory brainstem response looks pretty similar in humans and rodents. We can also measure the acoustic startle response, so the motor response to a sudden loud noise. In humans, we play sound and we normally measure the EMG from beneath the eye. So we basically measure the eye blink, which is part of a startle response. Whereas in animals, we normally measure the entire body, like the whole body flinch, by putting the animal into a little holder and playing the sound through a loudspeaker. But the startle response is not an all or none response. The startle response increases as we increase the sudden loud noise, and these input-output functions look pretty similar between humans and rats. Likewise, of course, we can ask a kid or a study participant to discriminate, for instance, loudness, what is loud, what is not so loud. And we know from other studies that mostly there's normally no impairment or no change in loudness discrimination. We did the same with our rats. It's a little bit more complicated in rats because we can't just instruct them to hit a key. We need to use operant conditioning. So we need to condition a rat to nose poke when something is quiet on the right side and to nose poke when something is loud on the left side. And when we do this and we train them with a loud and with a quiet sound, and then we provide sounds in between, we get this nice psychometric function that is the percent of the stimuli, the respective stimuli, this is the loudness here, the volume, that are rated as loud. So the quiet ones are always quiet, the loud ones are always loud, and the interim stimuli are always somewhere in between. And likewise, our catnap knockout animals are not different from the wild type animals when we test them to rate the loudness. Lastly, another test that we did is sound avoidance. That's a little bit different because that is the subjective perception of sound. So not just rating it, but how annoying is sound. And we know from kids with autism or people with autism that they are often not very tolerant to loud sounds, that this can really trigger meltdowns, and they seem to be often more sensitive to sound. And so with the rats, to ask them how much do they actually mind the sound, we built this sound avoidance box where we put the rats into a dark box where they like to be, but they can escape through a tunnel into the light box where they don't like to be. They like the dark and cozy space. But then we put an increasingly loud noise into that dark box, but not into the light box. And then we see at what point the rats actually mind the noise and they prefer to go into the light box rather than staying in the noise in the noisy surroundings. And when we do this, we see that in blue is the wild type, and in orange is the knockouts. The green is the heterozygous, so they are often in between. But what we can see is that the catnap knockouts start to leave the dark box at relatively moderate sound levels already, whereas the wild type stay a little longer in the dark box with relatively loud sounds. They all escape when it's 83 decibel or louder. But in these decibel range, 65 to 74 decibels, we have a significant difference where we can see that the catnap knockout rats, when given the choice, prefer the non-noisy environment and they leave the box. They spend significantly less time in the dark box to avoid the noise level. So to summarize maybe 10 years of research, what we find in the catnap knockout rats and in humans is that the auditory brainstem response actually is lower and slower in children diagnosed with autism, and it's also lower and slower in juvenile knockout rats. But then as they approach adulthood in both the knockouts and the study participants with and without autism, in adulthood, there is no difference in ABR. So the ABR, the auditory brainstem response becomes faster and stronger during development. So when it's lower and slower in autism, that means that there's a delayed development of the ABR. We know that our auditory evoked potentials are higher in humans with ASD. We didn't measure that, but other people did. And we do see higher cortical activity, higher cortical excitability in our catnap knockout rats. When it comes to startle reactivity, the autistic group had an extremely much higher startle than neurotypical kids and adults. And we see a highly exaggerated startle response in the catnap knockout rats as well. When we look at objective loudness rating, as mentioned, there's no difference between autism and neurotypical, and there's no difference between catnap knockout and wild type. But when we look at sound aversion, so subjective sound processing, then there is a higher sound aversion in autism, and we see the higher sound aversion in our catnap knockout rats. So that is, a lot of work of a lot of students going into that. So a high level summary. So what I just summarized is that there is a delayed maturation of auditory processing in the brainstem. So this, what we've measured with the auditory brainstem response, this pathway is lower and slower. But at the same time, kids with autism and our rats develop this exaggerated startle response. So that is kind of interesting because the startle response, the input into the startle pathway is actually this first part of the auditory brainstem response. So we know that the input into the startle pathway is lower and slower. But the animals startle, and the kids as well, startle much more. So something must be happening in the reticular formation that makes them startle higher, although the input we know is actually lower. And this is, of course, where we can leverage the animal model and to really look what is happening there. We also know that they have intact loudness perception, so that is on the cortical level, but there is a different subjective perception of sound. So the value of sound is different in autism because there's more aversion to sound. So we can now leverage the animal model and ask, what are the cellular molecular mechanisms underlying these changes in sensory processing? Why do they startle more when the input is less? What happens in the cortex and so on? And then we can test interventions. And so the remainder of my talk, I want to briefly show how we measure these cellular and molecular changes and what interventions we test. We have drug interventions, but we also look at sensory enrichment and basically something that is non-pharmaceutical, something we could probably compare a little to behavioral therapy. And we look at this as an acute intervention, but also, does it have lasting effects when we do this early in development? What are the long-term impact when we do this during the critical period of sensory development? And I want to show just a little bit, some snippets of data, because I'm aware I will otherwise run out of time, but just to show you how we approach this. So again, here is the startle pathway, and the input into the startle pathway is the spiral ganglion cells and the cochlear root neurons, which are these neurons here that we actually measure with the ABR. They are lower and slower early in development. But then something must happen in the PNC, in these premotor neurons that directly project to the spinal cord, that actually lets the animals startle higher, and that the animals, catnap knockout animals, startle higher. We've shown over and over again. Kayla showed that in her thesis. Dorit, a postdoc, showed it. And Ala Sheikh Mohammed, another PhD student in my lab, showed that over and over again as well. So we look a little bit closer at this reticular formation at these PNC giant neurons. So Alice Zhang, another MD PhD student in my lab, she graduated last summer. She put these rats into a stereotaxic frame and then lowered an electrode into the PNC, into the reticular formation, and measured the response of these neurons to sound. And what she could see is that these are the wild type. This is the firing frequency of the neurons. This is different sound levels. And we can see that the wild type fire at a specific frequency, but the knockouts in orange, they fire on a much higher frequency. This is specifically true for the females, which are the dark colors. The males are the light colors, so the knockout is a little higher, but by far not as much as in the females. And they also have a lower firing threshold, so they fire at lower intensities as well. And they are, she also looked at expression of an immediate early gene. So she played the startle response and then looked how many of these PNC giant neurons, startle neurons, were actually activated. And she could also show not only do they fire in a higher frequency, there are also more neurons activated in the knockout animals than in the wild type. So they are more likely to get activated. And the number of neurons that get activated does actually correlate with the amplitude of the startle response of the respective animals before she got the brain out. So that is one of the things we did. So we see the higher excitability of these startle neurons in the reticular formation and a higher recruitment of these PNC giant neurons, specifically in male knockout rats, whereas in females, there's more this higher excitability. We then, another PhD student in my lab, Raj Kamal, who also graduated last summer, she then takes, the brains of the knockout rats, takes slices out. And on these slices, she has this part of the startle pathway. We have another input here, which is the tactile input, but we look mostly on the auditory input. And in this brain slice, she can then record from these startle-mediating neurons the tiny little postsynaptic potentials and action potentials while she stimulates the auditory input. And so what she found, and again, without going into much detail, but that in wild types, the females and the male, this postsynaptic response, the electrical response in these PNC giant neurons normally go down upon repeated stimulation, but they don't do that in catnap knockout rats. So they seem to stay up high all the time. And again, so that shows that they are more excitable. They stay also more excitable, these neurons in the PNC. And of course, this is, again, only a snippet. We now can perfuse all kinds of ion channel blockers and synaptic blockers and can find out why that is, what is actually the mechanism that makes them more excitable. Dorit Merle, who was a postdoc in my lab, she also looked at the PNC in terms of neurochemistry. So she used a technique that is called multi-TOF, which is a mass spectrometry for every single pixel of a brain slice on a slide. So every single pixel is a full mass spectrometry for small molecules. And so when she zooms into the PNC, where these startle neurons are located, she looked at specifically 3 different peaks, which are the peaks for glutamine, glutamate, and GABA. Glutamine is the precursor for glutamate, which is the major excitatory neurotransmitter. And glutamate is actually a precursor for gamma aminobutyric acid, which is the major inhibitory neurotransmitter. And as we all know, or many of you hopefully know, that there's always this theory that in autism, the balance between glutamate and GABA, between inhibition and excitation, is a little off, which leads to this hyper-excitation. And what she found is actually that all 3 of them are totally off in the knockout rat, that all 3 of them are increased. So something is up with synaptic connections and with neurotransmitter release in the PNC. And that's all I'm going to show for the PNC. I'm going to show one more data slide about the auditory cortex, which is related to this more perceptual processing of sound. And what Kayla Scott, she again recorded in the cortex in vivo in the catnap2 knockout rats. Raj Kamal did patch clamp recordings and brain slices, but the work of both of them show that that these PNC, these cortical neurons, the pyramidal neurons in the auditory cortex, fire more, they fire longer in response to a sound stimulus. So there's a longer duration and a higher firing rate in vivo. And when we look at the brain slices, we see that the firing threshold is lower. So that means they are more easily excitable. And the Rio base, which is the current needed to make a single neuron fire an action potential, is less in the knockout. Again, a more excitable neuron. Okay, so what we have shown is that we have intact loudness perception, but different subjective perception of sound in the catnap knockout rats, paralleling the situation in autism. And this is paralleled by a higher excitability in the auditory cortex. And the question is, of course, is that a compensation because the input is lower and slower during development? Or is this a direct effect of catnap2 knockout that, you know, there is some problems with synapse stabilization or so in the auditory cortex as well? I also showed that there's this delayed maturation of the auditory processing in the brainstem. So the auditory brainstem responses are lower and slower, but we still have a higher startle reactivity despite this lower ABR waves, despite a lower input into the startle pathway. So I want to emphasize, so these rats and kids with autism are obviously not hypersensitive to sound. They are actually hyperreactive to sound because the auditory brainstem waves show that they are actually less sensitive, but they react more to sound and they find the sound much more annoying. And again, the big question is this a direct catnap effect, or to what extent is this a compensation of the brain to this lower and slower input that it receives during the critical period of sensory development? And so the last 5 minutes or so, I want to elaborate on this hypothesis, whether there is a compensation for this delayed auditory brainstem development. And I want to also talk a little bit about potential treatments. So one of the drugs that we looked at is baclofen, arbaclofen, which is a GABA-B agonist. It's used to treat muscle spasm after injury, and also sometimes is used off-label for withdrawal symptoms. Arbaclofen has been, there has been huge clinical trials with arbaclofen as well. Many of them failed utterly for different reasons. I think one of them I mentioned before is, of course, the participants diagnosed with autism are not catnap2 rats, or are not similar as catnap2 rats, because they might have very different causes for autism, whereas in catnap2 we know there's one single cause that is, you know, representative of, I don't know, maybe 5% or maybe 10% of autism cases if we talk of the entire cluster, that the pathway that is impacted. So that is one problem, but it's also a problem, of course, that the readouts of this trial relied heavily on parent questionnaires, and there were other, so it was hampered with all kinds of difficulties. And it failed, but the Simons Foundation for Autism Research wanted people to revisit baclofen, and so they provided arbaclofen to researchers and clinical trials, and so we got arbaclofen to test it in our rats. And Dorit Merle did that work, and again, I don't want to go too much into detail here, but these are startle responses in females and males, in the wild type and in the knockout rats, and we use different doses of arbaclofen, and we saw a concentration-dependent decrease in startle response in both, actually, in the wild type and in the knockout animals. So arbaclofen partly reverses the increased startle responses in catnap2 rats. It reverses partly other symptoms as well, but not all of them. And it does have, we have to be very careful of motor effects, direct motor effects of the arbaclofen as well, of course. So what we do now, after this first proof of principle test, is we try early treatment. So what we are currently doing, and I'm not going to show you data, we have only preliminary data. It looks very promising, but it's not at the stage I want to show it. But Ella, a PhD student in my lab, together with an former undergraduate student, Gurvinder, in my lab. So what they did is they inject arbaclofen daily from postnatal day 14 to 21. And that is the critical period for auditory development. This is where this hyperplasticity occurs, where the brainstem is low and slow in the catnap2 animals. And so they inject arbaclofen for this 1 week. But then they test the animals after they have ended the arbaclofen treatment as juveniles and as adults. And they do the entire behavioral testing battery. So they look at startle and prepulse inhibition, social behavior, locomotion, and so on. And not only that, I have Hannah, well, she still is a master, a PhD student to be in my lab, who does the electrophysiology, the patch clamp recordings, then in the slices of animals that have been injected during the critical period to see how this treatment in the critical period changes maybe the excitability phenotype in the catnap knockout rats. And then Renee and Cleusa, Renee is a third-year undergraduate student, and Cleusa, my lab tech, they do Western blot. We don't do MALDI-TOF anymore, but we do good old Western blots to actually look at the content of GABA and glutamate and glutamine in these brains of the treated rats. And of course, we have a control group that are injected with saline instead of arbaclofen for comparison. And one last slide I want to show is, so this is a drug intervention, but I mentioned at the beginning that we also look at basically behavioral interventions. And so one intervention that we are looking at, and others have before us and have shown some effect as well, is sensory enrichment. So we raise rats, wild type and knockout, either in standard caging, which are very bare, small shoebox cages, or we raise them in these enriched cages, which are multi-tier, and they can climb up and down. There are a lot of toys. Many of them make sounds, and the animals do interact with them. They have a running wheel. They have hammocks, which are face masks. So there's a lot of stimulation. We raise entire litters in that environment. So these are very young ones. They probably can't hear yet, but during the critical period, so as soon as they start hearing, they are in this environment. We raise them there, and then after the critical period, we put them in standard caging and compare them with animals, knockout and wild type, that are raised in the standard environment. Yeah, and with that, I want to conclude. So what I hope I showed you is that there are changes in auditory processing in these catnap2 rats that really mirror changes that we see in autism. And actually in a large population of autistic individuals. That these sensory processing disruptions in autism offer a unique opportunity for translational studies because the pathways are so similar. But then, of course, we can use the animal model to drill into the mechanisms, to really do invasive things like electrophysiology and Western blotting and things like that, where we can take the brains out and drill down to the mechanisms, and we can test all types of treatments. And with that, I want to mention the collaborators on this work: Brian Allman from my department, who is a specialist in vivo electrophysiology; Ryan Stevenson from the psychology department, who is the one responsible for everything in humans; Janice Carley from the National Center for Audiology, who helped us setting up the auditory brainstem response recording; and of course, Rob, who helped us with patients, with
Oh.
participants. Sorry, I was distracted. And with that, yeah, I want to acknowledge, of course, all the students, former and current students in my lab, and all the funding agencies that supported this work over the last years. That's it.
Suzanne, thank you. As always, that was fantastic. If I may say so, that picture of me was 20 years old, and although it is nice to reminisce, it's also a bit disappointing to see what has happened to my hair over those 20 years. So, well, thank you, but not thank you at all. So if I can ask first, you know, actually, if I can just say, so if people have questions, I think we would prefer it if people use the raised hand function, just because that way people, if they want to, can ask questions through audio, and we can hear them. But I don't know oh, there we go. But if otherwise, people could also use the chat function, I suppose. But with regards to treatment stuff, so where do you see this research going in the future?
A good question. So I think for now, we really want to see what these early interventions do. As always, from my perspective, we find something out, we have a result, and it always opens up 10 more questions. So if we have some preliminary data that show that there are lasting changes when we do these early treatments, which and probably both, so the sensory enrichment as well as the drug changes, which I think is also promising. But of course, we need to know, we actually would like to know what kind of changes. Again, we want to drill down on the mechanisms, like what can we actually change and what can we not change with these early interventions. And I think in the future, it would be great to really look into translation, because I think it does have implications for treatments. What can be done by early interventions, like what can be changed, what cannot be changed, drugs versus non-drugs. But we also always need to keep the limitations in mind. These are catnap2 knockout rats, right? So this have a very specific biological cause. And it whatever, especially the drug treatment, it might help in the catnap2 rats, and it might help in, SHANK3 and genes that interact with catnap2, but it might not be valid or efficient in autism that is not related to these genes, but have where a different biological mechanism is impacted. So I hope that the field, we can't do that alone, but that the field is moving a little bit more into differentiating within autism to what are actually the impacted biological processes. It is very, in every other disease that is not neuropsychiatric, that is not connected to the brain, the diseases are more defined by biological disruptions. In the brain, it's not, of course not, because we don't have the same access, and we don't understand the brain in the same way we understand the heart or joints and things like that, or the pancreas. But I think with the development of techniques, we might be able to get a little bit more into that direction. And so as much as this is a development in cancer and in other disorders, I think there will never be the golden pill that cures autism. We will have to have more information about the cause and probably more of a personalized approach.
But no, I agree with 100%, because I think unless we have the phenotype down really solidly, it's going to be difficult to have any research that really makes a difference in the long run. But I think also this kind of work you're doing though is great because it based on, so I think you know that I was involved in a clinical trial of our baclofen.
Yeah.
And I think what we found was, and I think the sample size at a number of sites in Canada, the United States, or in Europe was, I think our sample's 220, which isn't big, but it's big in this population. And I think what was found was that parents rated their kids, and it was placebo controlled. Parents of people taking our baclofen rated greater improvements in socialization and social interactions and engagement and things, but it didn't reach statistical significance, which is what I said, God loves 0.06 as much as 0.05 probably. But I think that's the standard that we set. And I think that one of the problems we have in humans, really, that again in animals you wouldn't have, is outcome measures. So how
Yeah.
do you really measure social interaction? It's a very difficult thing to do. And I would imagine in rats, it's a lot easier to do those kind of things.
Yeah, I mean, we have, of course, standard tests that are, a little limited as well. There's a standard test, it's called a three-chamber social interaction, which is the one we used. And when I say, novel techniques, so there are novel techniques emerging there as well that could be potentially even used in humans. And that comes with AI. So where you actually just videotape rats in their normal environment, and you could basically videotape a kid, interacting like in a room with others, and use AI to then rate social interactions. So these new techniques that emerge are, I think, have the potential to be much more sensitive, still objective because it's AI, right? So it's not a subjective rating, which is always also a problem. It is an algorithm that actually analyzes the data. You have to train it, of course, so that is always a downside. But I think these novel techniques might actually increase translation as well. So that is something that we are looking at to actually change how we rate social interactions by not these standard tests where an experimenter takes the rat out the cage and into a maze. That has all kinds of issues, because if catnap rats are more anxious, then they might react differently to that manipulation already, right? And that might impact their social behavior as well. So having this more naturalistic behavior, that is one thing where the field goes to. But I think there's also a potential to maybe even use this for human measures that are a little bit more objective than parent questionnaires, which are also very much influenced by the stress level of the parents and their perception and things like that.
Oh, and almost certainly by recency biases and stuff like that, right?
Yeah.
And I think that in fact, what has been shown in ADHD and autism more recently is if you give parents a standard rating scale, do nothing, and then ask them to fill it out 2 weeks later, and scores tend to go down. And so I think that, how obviously what placebo responses and stuff like that we need to consider. So Dr. Claire Mitchell had a question out of here that you mentioned that the mice and individuals with ASD may not be more sensitive to sound, but are more reactive to sound. So the question was, do you think this is true for most patients with ASD, and what might be the implications for treatment? And so, and my addition to that would be, and I've wondered, I think we've talked about, how much is an excessive startle response, and what influence does that have on people?
Mm-hmm. Yeah, so that is something we measured. So the participants did have autistic participants did have a higher startle response than neurotypicals. So that is the same situation in humans than in our rats. By the way, it's not mice, it's rats. I want to emphasize this. In terms of social behavior, it makes a big difference. But also we see the same situation with the ABRs. So the ABRs are lower, but the startle is higher. And ALA did actually measure this in the same individuals. And it is not, that we have this, it's a huge effect in the catnap2 rats, whereas the human population is much more diverse. Of course, again, they are not all catnap2 knockout humans, right? But we do see the same effect in humans.
So, but if the issue is that people with autism overreact or are excessively reactive to sounds or whatever, as opposed to are overly sensitive, can you speculate on what the treatment implications of that might be?
Yeah, and that's, I think, is also important, because if that is true, and it seems to be true, at least for auditory, to maybe they should be exposed to sound, or maybe we can, so that if this is really, and that is our early interventions right now, if this is a compensatory mechanism in the cortex and in the PNC that seems to compensate for the lower input. So what we think is happening, so maybe there is lower and slower inputs from the brainstem into the startle pathway, into the cortex, early in development because of the delayed auditory brainstem maturation. And so the cortex and the PNC compensate for that by upscaling the amplification, basically. So they basically amplify more the signals. So because the brain always wants to have this optimum level of activity, right? So they amplify the signal, but then during the critical period, but then they are stuck with this amplified signal. And so the auditory brainstem eventually catches up and is normal during adulthood. But now they are stuck with this amplified signal, which makes them, more reactive to sound and makes them hyper, I don't want to say hypersensitive, because the sensory pathway in the periphery is not hyper, but this amplification along the sensory pathways then more centrally, they're stuck with that. And so maybe, that means if that is true, and we can test this with our rats with these early interventions, that means we should try to counteract this hyposensitivity, actually, that makes them hyper reactive. In some way, we need to find maybe a way, or maybe we have to expose them more to sound early on. Maybe that helps. That is our environmental study, right? So maybe just exposing them more, not less, but more, would help, which is also easier said than done.
But that's a really interesting way of looking at it, because I think parents go out of their way to shield their kids from noise. So
Yeah.
you see all these people walking around with headphones on all day, and it's a very different way of looking at it. Actually, Claire, did you want to say anything about that?
Yeah, I just think it's a fascinating finding, and that may well have some implications, because often our treatment is to try to, change the environment around a child, for example. So if they do appear to be hypersensitive to certain things, we might remove some of that from the environment. But what you're saying is perhaps we shouldn't do that, that we should continue to expose them and help with the reactivity side of things for the long term. And I'd be very interested actually to see what happens in the rats. And I'm sorry I said mice. I reread it and said rats. But anyway, if you do that in the rats, what that changes in terms of their longer term reactivity and your findings. It is a very different way of approaching it than we typically approach the young children at this point.
But Claire, don't you think, and I was just thinking, but I think it's the same with cognitive behavioral therapy for anxiety and exposure and response prevention, right? I think as parents, we go out of our way to protect our kids or rescue them if they're really upset. And I think we know with anxiety that exposure is really the most important treatment. And so it's just a bit counterintuitive. And I wonder how difficult that becomes for parents to do that.
And that's the other point, of course. Yeah. How difficult is it? Sorry.
But I think it's graduated, right? So I think, it's well described for anxiety. And I think the key is keeping it graduated so that each level is tolerable. And I just wonder a little bit about that for individuals with autism. And then I suppose we do it in a way by sending them to preschool, where they're going to be exposed to noise and chaos just naturally. And we know that helps. But anyway, it's interesting to kind of postulate about the mechanisms behind all this.
Well, and so Lisa Hoyland has raised her hand also. Lisa is an occupational therapist. Lisa, let me just see if I don't know if I can unmute you. But can you unmute yourself?
Maybe I should stop sharing.
No, I think there used to be, and I don't know if Jason would, because I think normally, Jason, would be an easy way for all I have to do is I could unmute people normally. Oh, there, Lisa's unmuted now.
Yeah, I think I've unmuted.
Yep.
Hi, yeah, the talk was very interesting. So I know in some of the occupational therapy research looking at sensory modulation challenges in children, there has been some research looking at outcomes where the children don't habituate to the same sensory stimuli. So in terms of looking at sort of a desensitization or an exposure type of model, they're thinking that kind of is not going to work because the children don't habituate to the same
Mhm.
stimuli that they're exposed to. And they did multiple things, not only auditory, but also tactile and vestibular input as well. So they're measuring some physiological reactions in the children. I'm also very interested in the sensory enriched environment. Essentially, that's what the sensory integration sort of treatment in theory is all about, and very interested in the fact that there's a vestibular input for the rats in those environments. In occupational therapy, we often think of the vestibular system as an overarching sensory system that impacts all the others. And again, going back to the fact that all of the sensory information is integrated and it kind of can't be looked at in isolation from all the other senses. So really looking at, again, at that proprioceptive and vestibular sensory systems that can be added to the typical 5 that we think about.
Yeah, I absolutely agree. And as Rob mentioned, I also study sensory filtering. So we actually do look at habituation and prepulse inhibition, which are these sensory filtering. And yeah, we see a lack of habituation in the rats, which of course, so they're not only changes in the startle, for instance, but they also do not habituate, which complicates things. I didn't go into that because it's a little more complicated to explain all the habituation and prepulse inhibition paradigms. There was just no time. The vestibular one is also very interesting. We did a little bit on audio visual integration in one of the studies, but one thing that I would really want to do, where we don't have the means yet, but we are applying for equipment to be able to do that, would be interesting also to monitor actually in our environmental enrichment cages how much the rats do interact and which rats interact how much with what type of toys. They do interact quite a bit. So we see them running in the running wheels and running and jumping around, especially the young ones. They are very active. But it would be interesting, of course, whether there are differences already there in the way they interact with the environment, and then to really look at the different type of stimuli as well. Unfortunately, we are not there yet, so we just throw them in there and then we see what the outcome is 3 weeks later. And yeah, and again, we do have only snippets of data so far. And at the moment, it looks like that the environmental enrichment changes some things and does not change other things. So yeah, that's all I can say at the moment. But I promise to come back in 2 years or so, and then I can report on that.
And we will, I think, hold you to that. But Lisa, around the question of environmental enrichment, because it's first of all nice to see people looking at other things besides just medication as ways of dealing with things. My understanding, Suzanne, I think you know more than I, but in people with, I think in Fragile X models, I think they've shown environmental enrichment can actually change sort of the neuronal structure later on, as a huge impact. And it's really nice to see that kind of work. And especially with Fragile X, we have it at a very basic science level, so the neurons look, I guess, more normal, for lack of a better, or I don't know what the technical phrase is.
Mhm.
It's really nice to think about that as other ways of doing stuff.
Yeah. Yeah, I agree. And I mean, a lot of that has been done in mice, at least. The problem is
So
that
I
mice.
will also apologize, just like Claire did. And I know that we discussed this once. And I know I said something about mice and rats being the same. And I remember you were quite upset about, but just honestly, I apologize ahead of time for getting
No
those
problem.
picked up, because it will happen for sure. But but it's nice to consider how we can do stuff with environmental stuff, because it's also a more, I don't know, ecologically valid approach, I suppose, in a lot of ways.
I absolutely agree. And I mean, the hesitance to drug small, I was actually pretty upset to hear from you last time how much and how very young kids get drugged already. It's somehow counter, and I don't know, it's something I can understand that that parents are a little hesitant to give very young kids drugs that
Many
tropic
parents are.
drugs that interfere with brain development.
Well, that's, I mean, so many parents are, some aren't though. And that, I
Yeah.
think, is the problem, right?
Well, it also is different levels of desperation, maybe sometimes.
Well, absolutely. And I think it's also, as a profession, I think psychiatrists want to sell you the idea that medicines treat everything,
Yeah.
and they can't. Are there any other questions or comments for Dr. Schmidt? Okay, well, so Suzanne, again, thank you very much. I appreciate it. It's wonderful to hear again. If I encounter those patients that we met before, I will let you know if they're coming, and we can sort of figure out if you want to see them or something.
Yeah,
Or
absolutely.
students.
Yeah.
But so again, thank you, Suzanne. I think Jason just had a comment in the chat about please, there's a link there for people to follow regarding the evaluation and any ideas for future. And also, I think Jason said here that he will email it to people as well as their certificate of attendance for continuing education credits. So in next month, I think it's January 8th, if I'm not mistaken, but if I am, Jason can correct me. I think our next speaker actually is, it is January 8th, is Ryan Stevenson. Actually, I see him talking about sensory issues applied not in sort of animal models, but in human models, we'll call them. So hopefully it's a nice follow-up to Suzanne's talk. So thank you again, Suzanne. I really appreciate it a lot. It was great to see you, and we will see everybody in a month.
Yeah. Thanks for having me. Bye.
Thank you very much, Suzanne. Take care.